All right, everyone, it's the top of the hour. We'll get started. I'd like to welcome everyone to today's GlomCon. Today is one of our clinical trials, conference series, which is a joint venture between GlomCon and NephCure. And today's title is APOL1, a story from bench to bedside. So before we begin, I just want to bring up NephCure, our partner in this series, where their mission is to accelerate research and effective treatments for rare forms of nephrotic syndrome and to educate and support to improve the lives of those affected by protein-spilling kidney disease. One of their resources they have is the Kidney Health Gateway, where physicians, patients, family members can look for clinical trials that may be going on in their area to help us get patients into trials, patients to participate in trials, and advance our understanding and treatment for our patients and their kidney disease. So today we are excited to have our speakers and panelists with us. Our speaker today is Dr. Stacy Johnson. She is on the clinical development team at Vertex Pharmaceuticals, working to develop therapeutic interventions for APOL1 mediated kidney disease. She received her MD and PhD from Baylor College of Medicine in Houston and completed her internal medicine residency and nephrology fellowship at Duke University. Dr. Johnson was most recently an assistant professor at the University of Tennessee Health Science Center in Memphis. And in June this year, she left to join the APOL1 mediated kidney disease team and lead here at Vertex. Our panelists today are Dr. Suzanne Nichols. She is a tenured professor of medicine at the David Geffen School of Medicine at UCLA. She received her medical degree from the University of California, San Diego, master's in public health from San Diego State University, and a PhD from the University of California, Los Angeles. Her primary research interests are to understand and identify key factors to promote the pathogenic medicines of diabetic kidney disease, uncover and validate novel therapeutic targets, and predictive biomarkers of diabetic kidney disease, and also perform population studies and disparities in chronic kidney disease and CKD health. She has served numerous leadership positions, both locally, nationally, and internationally, has numerous publications, and has served as mentorship for many, and received many awards for all of her work that she's done. So we're excited to have her here. Our other panelist is Dr. Jeffrey Kopp. He is the chief of kidney disease branch of the National Institute of Diabetes, Digestive, and Kidney Disease in Bethesda, Maryland. Was educated at Harvard and the University of Pennsylvania Medical School and received his training at the University of Washington. His research interest lies in the pathogenesis and treatment of FSGS, and his genetic studies have contributed to the identification of APOL1 as a major susceptibility gene for kidney disease in African Americans. He has numerous publications and holds a commission in the United States Public Health Service with the rank of captain. I'm Dia Waguespack at McGovern Medical School in Houston, and I will be moderating along with Joshua Tarnoff, our the Chief Executive Officer of NephCure Kidney International. So with all of that, we're excited to have everyone here today. We will get started. We'll start with Dr. Johnson. We'll do a presentation, and then we encourage everyone to ask questions in the chat, and we'll have a discussion afterwards for the remainder of the session. So with that, I will stop sharing, and Dr. Johnson will let you share your screen.
Okay, thank you very much, Thea. Okay, let me go to the slideshow. Okay, you guys see the slide?
Yes.
Perfect, thank you.
Looks good, thank you.
Hello, everyone. I know for some of you it's the morning, and for some of you it's the afternoon, so I'll say good day. And thank you so much for allowing me to be a part of this panel and to tell you a little bit about what's happening with APOL1 mediated disease at Vertex. For today's talk, I'll go over some of the genetics and mechanisms of disease of APOL1 mediated kidney disease, or AMKD. I'll also talk about the spectrum of clinical presentation for APOL1 mediated kidney disease and its clinical impact, and address the importance of genetic testing for APOL1, and also talk a little bit about Vertex's APOL1 screening study, and also give you guys an update on where we are on the pivotal clinical development stage of inaxaplane, which is a small molecule designed to inhibit APOL1 mediated toxicity. This is an investigative product, so thus far we're still early in the development, and no drug has been approved or has been proven safe for use. But Vertex is hoping to be on the leading edge of this. So I don't have to tell the people in this audience, but as we know, we have observed for many years a racial disparity in the incidence of end-stage kidney disease in the United States. Data from the U.S. Renal Data System, or USRDS, shows this disparity quite clearly, where people of African Americans or Black race in the United States have much higher incidence of end-stage kidney disease compared to other ethnicities. It was felt for some time that there may have been a genetic component to this, but for many years we lacked the tools to be able to determine the genetic basis. Since the 1950s, genetic gene mapping has come a long way, and with the development of improved technologies and statistical methods, we have been able to make a lot of strides in our understanding of some of the chronic kidney disease. In particular, in 2010, Pollock and his group identified APOL1 as the gene responsible for increased risk of kidney disease that had previously been detected on chromosome 22. Although APOL1, at the time that this gene was identified, its role in the kidney was not well defined. Since then, we've obviously had a lot of people contribute to our understanding of APOL1, and we know that the gene itself, the APOL1 gene product, seems to protect against the human African trypanosomiasis, or sleeping sickness. This infection is caused by the Trypanosoma brucei parasite, which is carried in the tsetse fly, particularly found in sub-Saharan Africa. The G1 and G2 variants of APOL1 are believed to have evolved in sub-Saharan Africa to overcome the ways in which the trypanosome is able to evade cell mediated lysis. And so what we see is that these variants are found predominantly in people of recent African ancestry, and individuals with this type of variants could be found in sub-Saharan Africa, but also in North America, Latin America, and the Caribbean, as well as some parts of Europe. And we now know that the presence of two of these APOL1 risk variants, labeled here as G1G1 or G2G2 or G1G2, have been linked to increased risk of kidney disease. The expression of the APOL1 risk variants may result in toxic gain of function that leads to cytotoxicity in kidney cells. The presence of the variant itself, not all individuals who have these risk variants will develop kidney disease, and it is felt that there is most patients who develop kidney disease with these variants often suffer a second hit caused by inflammatory or infectious mediators that lead to cytotoxicity. The cellular injury that's driven by APOL1 variants is thought to be the common mechanism leading to this progressive glomerular dysfunction and proteinuria, and it can be seen over multiple clinically and histologically defined kidney diseases. This slide shows you some of the clinical impact of APOL1 mediated kidney disease and across a spectrum of clinical presentations. We know that, as you can see here, particularly patients with HIV, idiopathic FSGS, and hypertension, attributed end-stage kidney disease, there's a the individuals that have two of the risk variants have a much higher chance of developing end-stage kidney disease. And this received, in addition to the program, this shows you here with, if you look at cohorts, FSGS, as well as hypertension-associated kidney disease, and even in the setting of membranous nephropathy, patients with AMKD and the two risk variants progress to end-stage kidney disease faster than patients with either no risk variants or only one. The clinical toll, or the toll of AMKD is particularly, the impact is particularly severe because it tends to affect people in the prime of life, often with age of onset below age 50. We've often seen, you can also sometimes see adolescents and young people being affected by the disease. Often it's asymptomatic until the kidney disease is quite advanced. To date, there are no targeted therapies for AMKD, and most patients will be managed with antihypertensives, particular ACE inhibitors and angiotensin receptor blockers form the mainstay, since most of these patients have proteinuria. In the case of FSGS, they may be on steroids or calcineurin inhibitors, and some may require diuretics depending on the stage of their kidney disease. So this shows you here that there is a great unmet need, essentially, for targeted therapies that attack or deal with the underlying cause of the chronic kidney disease. Genetic testing, when we talk about APOL1 mediated kidney disease, genetic testing is something that often comes up. And currently, genetic testing is not widely available in the clinical setting. But if you think about who should be tested for APOL1 variants, current recommendations, you could think about patients who have FSGS or steroid-resistant nephrotic syndrome, patients or individuals who are being considered for living kidney donation, since patients who receive an allograft from a donor with risk variants for APOL1 actually have a decreased allograft survival. So would suggest that the toxicity actually is with the tissue expression of APOL1 and not the circulating levels. Consider testing people who have end-stage kidney disease, proteinuric, non-diabetic kidney disease before age 50. And then there might be other patients with CKD and proteinuria where there might be other, where you suspect APOL1 involvement, for example, in patients with sickle cell disease. The benefits of genetic testing, it will help us understand physician, patients, and as a group, we may be able to better understand the etiology of kidney disease, could predict give some prognostic information and help us predict the course of the kidney disease, and maybe even be informative for transplant decisions, as well as for patients who have a family history of kidney disease. Knowing the genetic, whether someone has the two risk variants may lead to institution of preventative measures more quickly, and it may even help ultimately identify patients who can be enrolled in clinical trials as we try to develop targeted therapeutics. And so on that note, I want to mention here that Vertex is actually conducting an investigational APOL1 screening study. The objective of our study is to determine the prevalence of APOL1 mutations in individuals with non-diabetic chronic kidney disease. Individuals of African ancestry or geographic origin, and that could include someone who was self-identifies as Black, Afro-Caribbean, African American, someone from Sub-Saharan Africa, or Latinx, which we define as someone which is defined as someone from Cuban, Mexican, Puerto Rican, or Central South America. Anyone aged 12 to 60 inclusive is eligible for the investigational study. And patients with a diagnosis of FSGS or other forms of proteinuria kidney disease not attributable to some other underlying cause, such as diabetes, and obviously patients would have to be willing to comply with the testing requirements, and the testing requirements requires one visit for a collection of blood and saliva. The second effort that Vertex is making in our goal to address this unmet need in AMKD is to we now have a small molecule, an inaxaplin. You may have seen it referred to or heard it referred to before as VX147. And our small molecule is now advanced to a pivotal stage of investigation. Vertex is currently working on starting up a phase 2 and phase 3 adaptive study. It's a double-blind, placebo-controlled trial for patients with AMKD. And the goal of the trial is to evaluate the efficacy, safety, tolerability, and the pharmacokinetics of inaxaplin versus standard of care. Key inclusion criteria, again, we're looking for adults aged 18 to 60 years old. Subjects should have 2 of the APOL1 risk variants. Proteinuria cutoff here is less than or equal to 0.7 up to less than 10 grams. And GFR spans a pretty wide range, 25 to below 75. And patients should be on stable standard of care treatment, ACE inhibitors, ARBs, etc. Exclusion, diabetes, uncontrolled hypertension, and known underlying cause of kidney disease, such as IgA nephropathy, membranous monoclonal gammopathy. For now, the study is focusing mostly on patients with FSGS, as well as proteinuric kidney disease, who may also have hypertensive kidney disease, but with well-controlled blood pressure. We have 2 phases, phase 2 and 3. The first phase is the dose-finding phase, where we hope to recruit about 66 patients. They will receive 2 doses of inaxaplin. Each group will be testing 2 doses of inaxaplin. And then after the phase 2, after 12 weeks, the phase 3 of the study will begin, in which case patients will then be randomized to either inaxaplin, a single dose, versus placebo. There is an interim analysis that's planned for 48 weeks, and we are looking at primary endpoints are going to be this change in PCR from baseline, the eGFR slope from baseline to week 48, but also we're going to do an analysis at the end of the trial, which is at the 2-year mark. Secondary endpoints for the study include the time to a composite outcome of a sustained decline in GFR of greater than 30%, onset of end-stage kidney disease or death. And of course, we're also going to be looking at safety, tolerability, and the pharmacokinetics of the small molecule. And inaxaplin is still very much in the investigational stage, but I did want to put this out there to let you guys know what's happening at Vertex with respect to APOL1-mediated kidney disease. If you want to find out more about the studies, I direct you to, we have 2 websites. Since the studies are currently recruiting within the US, we have for the screening study, the genotyping study, we have, it's APOL1program.com. And then for the inaxaplin phase 2/3 trial, it's called the Amplitude study. And so patients can go to amplitudestudy.com. Healthcare professionals, you can get some more information on Amplitude Study -ATP, which is more geared towards healthcare professionals as opposed to patients. And with that, I will stop there. Well, before that, let me just summarize what I told you. So essentially, what we briefly discussed this morning, APOL1 variants, risk variants are found worldwide. We predominantly see in individuals of recent African ancestry. Individuals with 2 APOL1 risk variants have increased risk of chronic kidney disease and a much more rapid progression to end-stage kidney disease compared to those with only 1 or no risk variants. APOL1 mediated kidney disease has serious various clinical manifestations and can often affects people in the prime of their life. Genetic testing is essential for diagnosis and optimal disease management. And at Vertex, we hope that our screening study will provide an opportunity for patients to actually get some of the genetic testing. And currently, we are about to enter phase 2 and 3 trial of an investigational therapy called inaxaplin, which is aimed at actually treating the underlying cause of APOL1 mediated kidney disease.
Thank you, Dr. Johnson. And so now we will, I think that this is exciting. I think as we're here to talk about today, I think APOL1, I think can really in the future, we're hoping to transform how we provide care to a large portion of our patients who have kidney disease, who we haven't had much to offer and in the future. And I think in one of the comments in the chat was on the who to screen and the hypertension. And I think that's the that diagnosis, that really what it is. So I think I'd like to start with talking a little bit about who we should be genotyping, where you see the future and implementation of this, and then maybe go on to some of the questions with the therapeutics and could our participants also please put in some questions in the chat. So if we can just start with this trial of enrolling patients for genotyping is exciting. In the community setting, and then even worldwide, as we have a global audience, where do you see how each of you should individualize our practice of who we should be offering this therapy to, and then what kind of structure do we need in place to be able to really give the full benefit of, we can identify a gene, but do we fully understand, or how can we counsel our patients on what that means? So if Dr. Johnson, do you want to start with some of your practice, and then we can ask Dr. Nichols and go from there?
So, as I said, genetic testing currently is not widely available, and that's, I think, partly due to 2 reasons. One is the cost. Typically costs maybe $400 or $500 was the last price that I heard quoted, not covered by insurance. And then, we still don't know, we don't have a targeted therapy to offer someone. And so the cost versus the benefit of knowing the genotype is still something that we have to answer. But, we hope that if we are successful in our endeavor to get us a safe and effective treatment that targets APOL1 mediated kidney disease specifically, then I can see genotyping being used to help determine which patients would benefit from a therapy like that. Right now, we kind of lump everyone and say, well, you have hypertension, CKD, so it just is hypertensive kidney disease. But maybe in the future, we would have patients who, in addition to standard of care, would get something that actually targets APOL1. That's how I see it being used.
Yeah, I can jump in as well. I think one of the things that we should reiterate is in terms of who are the individuals who are at risk. And as Dr. Johnson indicated, the onset, it's, individuals who are of African ancestry. In this country, however, there's a tendency to equate African ancestry with Black race. And I think that that's something that we should make very clear, that it's not someone who looks like me or like Dr. Johnson, for example, should be the only people who should be targeted for considerations for genetic testing. As Dr. Johnson also indicated, individuals of African ancestry are identified in other countries, in the Caribbean, in South America, in Central America, and they may be of Hispanic, self-reported or self-identified as Hispanic, rather than self-identified as Black race or African American ethnicity. And so they need to be also included in that target population. And I think it's been mentioned, but I want to be certain that it is emphasized so that these individuals do not fall through the cracks in terms of screening. Clearly, we don't have anything structured right now for screening because there are a number of issues that you need to take into consideration if you want to implement a sort of structured screening program. And as been mentioned, the cost. We have individuals where there's a huge disparity in chronic kidney disease incidence and prevalence, as well as in terms of progression to kidney failure and all of the cardiovascular risk in terms of mortality. We really need to consider as a population whether we want to advocate for coverage of screening for these individuals. So that's something that maybe we can talk about eventually, but it's certainly an issue. And also in terms of, who wants to know about the APOL1 test results, and if everyone wants to know, who's going to be providing that type of information, who's going to be responsible for actually activating and writing the order for the screening test and for delivering the results and how that should be discussed. So I think there are a number of issues that certainly have not been discussed widely that we need to discuss and address before we embark on implementing, some type of widespread screening, which I think is important and likely necessary.
Thank
So
you.
maybe I'll give Dr. Kopp an opportunity to respond. You're muted, Dr. Kopp.
Dr. Kopp, thank you for joining us. You can unmute and please feel free.
Thank you. Those are some great points that you just made. And I would hope that each nephrology division would already have or would identify somebody with a particular interest in genetics that would take this on and would learn about these various routes to genotyping. Certainly one is a whole exome scan. Another, in somebody who is self-identified as having African ancestry, would be to go straight to APOL1. But as I think you pointed out, we don't always know what our ancestry was. And really, if an effective therapy emerges, we will be wanting to test everybody. We're sort of in a gray zone now. We have some emerging therapies. We don't know which ones are going to be most successful. So I guess the bottom line is I would urge as widespread testing as is feasible, with a whole exome scan, with a targeted panel for FSGS, what have you, to try to identify people who may not realize that they potentially have APOL1 risk alleles. It'll become more pressing once we have proven effective therapies, but I think even at this stage, there's a great advantage to try to set our net as widely as possible to identify potential subjects.
And then Dr. Kopp, to that point, is there any utility in, there are particular patients who we treat on an individual basis, and our obvious goal is individualized care and precision medicine, where we can offer prognostic information when they are at a point of their disease where they're already progressing, not responding to therapy, to help guide them to things like transplant and a patient population who may not be as accessible, or we're trying to improve our access to those kind of things.
Well, you raised the issue of transplant, and certainly we would want to know if a potential donor had APOL1 risk alleles that have not yet caused disease. The donor has perfectly normal renal function, but might in the future. I don't know if that's the kind of issue that you were getting at, but certainly that is a concern.
And that is somewhere I think a field where more testing is happening is definitely center specific, but where the APOL1 testing, even for, like you said, donors and recipients, seems to be a bit more prevalent than it is on the general CKD population.
That's right. That's right.
All right, thank you. And then a question that has come up in the chat in relation to, the various phenotypic presentation of patients with APOL1. And if you can kind of comment if there's any thoughts on why mutations have a difference in their clinical presentation, and if we have any insight into that.
Well, maybe I can jump in. I don't think we have great insight, but it is so important to recognize that some patients with 2 APOL1 risk alleles present like primary FSGS with heavy proteinuria, low serum albumin, and others appeared like post-adaptive FSGS. And perhaps we can say that many of those would have risk factors like obesity, hypermuscularity, abnormal birth history with resulting fewer nephrons. So it seems to me that in many situations, APOL1 acts as an additional factor on top of other risk factors or other, maybe we should even say mechanisms of kidney disease. And so there are these different phenotypes, particularly primary FSGS versus post-adaptive FSGS, both of which APOL1 can either mimic or can synergize with.
And I guess, Dr. Kopp, that brings up the issue around that second hit, as you've mentioned, some of the other risk factors, but do we have any clear indication? There's some indication about inflammation, but for an individual, if I'm seeing my patient in clinic, what are the things I should be thinking about? Would you suggest that someone who says that they have African ancestry, maybe they have high blood pressure that's poorly controlled and they're advancing in kidney disease. What other things would you suggest that I would talk to this patient about in terms of alleviating their risk for progression, given that we don't yet have an FDA-approved therapy?
Well, I think it's the usual package that we all do. First, the non-medical things, weight loss if obese, stop exogenous androgens if somebody's a bodybuilder, exercise, diet, low salt, those things are all important. And then the usual things for FSGS or hypertension misattributed nephropathy. So ACE, ARB, low dose diuretic, sodium restricted diet, and so forth. So I think there's a package of things we already know have some benefit, but we have to acknowledge I've seen in my clinic, and I'm sure others have as well, patients who do everything they're asked to do on that list I just went through of lifestyle changes and taking medication, which are not easy, and yet they still progress. And APOL1 is clearly the driver for many patients. Now, there are many patients who are not of African descent who have similar kinds of stories. They do everything we tell them to do and they still progress. That's still a problem to be resolved. But now we have APOL1 as an explanation for some of our African descent patients, and now this promising idea of targeted therapy.
Thank you. And Dr. Nichols, I think, one of the things that we know is that there are great disparities in care. And with the, discovery of APOL1, the progress we're making, how do you see that fitting into being able to better, address these issues? And do you see that we can make some headway on this front?
I truly believe that we can make some headway. What I like about the knowledge we've gained around APOL1 is that it feeds directly into our ability and an opportunity to increase screening for chronic kidney disease in general. And as Dr. Kopp has described the risk factors that contribute to CKD in general, regardless of where your ancestry is, we're not doing a very good job around that globally. And for patients who are at highest risk, patients who are, the most vulnerable patients, who typically are patients that underrepresented minority groups, we need to really step up are screening for chronic kidney disease. And I think that with this APOL1 knowledge and the promise of new therapies on the horizon, that this may indeed be a stimulus for primary care physicians even to start thinking about screening for chronic kidney disease and trying to identify, what may be contributing to a patient's, if, for example, rapid decline in eGFR, as they're referring the patients to nephrology. I think it opens up so many doors for us in terms of increasing awareness and knowledge about chronic kidney disease in general, but obviously specifically about APOL1. And in terms of not just the genetic testing, knowing which test to order, as Dr. Kopp has described, some of the ones that are available, but who should those tests be ordered for? And once the results are back, who is responsible for delivering that information to the patient? How should that be done? And in terms of we as nephrologists, as well as healthcare providers, really need to be educated about genetic counseling. I don't think we do very well when it comes to genetic counseling in general. So maybe this is another opportunity for us to expand our knowledge base about that so we can impart that knowledge to our patients. So I think that this is really a great opportunity to help to reduce the disparities that we know have existed in chronic kidney disease for so many years.
And do you think that if we focus strongly on one thing that's a genetic variant, are we going to overlook other structural causes of disparities in healthcare? Is there a risk for that?
Well, I think there's always a risk, when we're looking at being too sort of narrow sided in looking at only one thing at the expense of others. So we have to be very cautious about that, knowing that APOL1 is one in the midst of several other potential etiologies for chronic kidney disease and that contribute to the disparities that we see. We talk about that second hit. We don't know what that is. We know that social determinants of health play a very important role as well in the disparities that we see in chronic kidney disease. And it's something that we really don't talk very much about, and so this is when we put a packet together to increase awareness and education for our patients, we need to have, I would say, first of all, a large descriptive of risk factors for CKD, what are the potential causes, et cetera, and then focusing on APOL1.
Thank you. And Dr. Johnson, I know before this session, we discussed a little bit about your work with an underserved population, and how do you see this fitting in, kind of as Dr. Nichols has already commented on, for those patients that you were serving, and I think probably part of your decision in your to change your role a bit to work on this more.
I have to echo everything that Dr. Nichols said. There are many patients who have chronic kidney disease and that has been, I think, misattributed to hypertension. And so as we start to, I think some of the same issues come up, of course, then we think about who to screen. And so some of these patients would be the types of patients that we would have to screen. But before we screen, we need to know, okay, what are we going to do with these results? How do we talk to the patients about what these results mean for them personally, but also for their relatives? And then, hopefully once we have safe and effective therapies, then how to get these patients to therapies. Because in some part, where in my work with patients in Memphis, again, with underserved populations, medication and therapeutic cost and access to therapies, especially new ones, will always be a barrier that then we have to make sure we have systems in place that we can overcome.
Thank you. And then, a question or comment that came up about developing countries where, you talked about the cost here and even lack of availability. What can we think about in the future? Can we even, think about this to be used and in those type of places as well?
I wish I had a good answer for that one, but yeah.
You know,
Yeah,
I
I think it's
would,
something to do that.
yeah, I would like to applaud Dr. Johnson actually, in terms of how you've switched, right? And now that you're able to get onto the other end to be able to bring therapies to patients. And I think that one of the things that I admire and one of the things that we should all work towards as well is increasing the workforce, right, in kidney disease for people who look like us, you know, so that patients who are vulnerable patients have a higher level of comfort in asking the right questions and in having that dialogue with their provider, something that we also need to work on in our, at least in our country. Yeah.
Thank you. And I'd like to go, there's been a couple of questions about the trial that you presented, and one particular question about inclusion criteria for the biopsy. Was it biopsy proven, those type of things? And then I wanted to talk, once we talk about that, if you can address that question, Dr. Johnson, then we will talk a little bit about the targets of these therapies, what Vertex is doing, and then maybe some others, and what we think, you know, this may lead to. And so the question was from Dr. Lau about clarifying who can enroll, and how do we, you know, find sites where those patients that may be eligible can go.
So the fastest and the fastest way to find the sites is so we have I talked about two studies. One was the genotype, what I'm going to colloquially refer to as the genotyping study, and the inclusion. So I would direct you to, I think it's the website is APOL1, and I'm going to have to go back to my cheat sheet to get that. But so look up the APOL1
program.com.
APOLprogram.com for the genotyping study. The inclusion there are adults 18 to 60 years old inclusive. The lower limit of with chronic kidney disease, the lower limit of proteinuria is, I believe, 0.2. So it casts a fairly wide net. The amplitude, it's if you look at amplitude.com, amplitudestudy.com, it will give you the inclusion and exclusion criteria for the patients who may be screened and eligible for the interventional trial. Currently, people with diabetes are explicitly excluded, which I know is a large population. So the focus is mainly on patients with FSGS. Yes, biopsy proven, as most of these patients will have a kidney biopsy showing primary FSGS, as well as those with hypertensive kidney disease.
Thank you. And then the investigative trial with the inaxaplin was an APOL1 inhibitor, correct? And so are there any, or do we know of issues with inhibition of APOL1? Are there any risks of neutralizing it? And do we have any information on that?
So I would think I'm not able to give any sort of preclinical data during this talk, but I would encourage people, if they have a specific question, to contact the Vertex Medical Information group directly, and that's their email address is medicalinfo@vertex.com, VRTX.com, and they can ask more specific questions. From that standpoint, I'm sorry, I can't give it.
That's all right.
Could I add a comment?
Yes, please.
Of course, if somebody lives in a region of sub-Saharan Africa where there's trypanosomal infection, inhibiting the function of APOL1 could potentially increase the risk for infection.
So Dr. Kopp, I have a question for you, because I know you've done all the research in this field. So APOL1 is not only, and many of the studies have been done looking at podocytes, knocking out the target signaling proteins within podocytes, but APOL1 is not purely expressed in podocytes. And in that regard, does that have any impact on, going back to the question in terms of potential side effects, if we're targeting, really it's primarily the kidney disease and proteinuria, would that have any impact?
Well, the major source of APOL1 is the liver, where it is put into HDL particles, and that is how it kills trypanosomes. To our knowledge, APOL1 is not required for normal function. And the evidence of that is somewhat fragile, but it was an individual in Africa who was identified as having 2 null alleles for APOL1 and appeared by evaluation to be completely healthy, but he became infected with a very unusual form of trypanosome. As I recall, it was trypanosome evansii that normally does not infect people because we have an effective APOL1 protection. That was the one phenotype that he had, that he developed this unusual infection. That's a pretty fragile read to say there cannot be a side effect of knocking down APOL1, but in general, I think we are hopeful that as long as you aren't at risk for African trypanosomes, that it should be safe to knock it down. But I think this study and others will be looking very carefully to see if something surprising emerges.
Thank you.
Thank you. And I think, Dr. Java had made some points about transplant and APOL1, and we kind of touched on that a little bit earlier. Can you comment, Dr. Nichols, about your experience with transplant, with what your center maybe does with screening, and then how that broadly affects to I think the disparities too in transplant access and all of that, and how you see that affecting our patients going forward?
Yes, sure. Thank you. That's a really important topic. So in our center, increasingly, we are doing more genetic screening testing in our patients, not only in recipients, but also particularly in donors, just based on the data that Dr. Johnson had described earlier, in that you may not have CKD, but having the risk alleles will put you at risk. And so for someone, particularly younger individuals who are coming in as potential donors, but maybe belong to have a family history of CKD, then it becomes really important. So we're offering the test, and of course it's somewhat expensive. So it is about $400, and it's not currently covered by insurance. I know for example, in cancer, if a provider orders a genetic testing, some of it can be covered. I'm not sure that occurs within our field. And if anyone knows that or has other experience, it would be good for us to know as well. But it's certainly something that we need to incorporate in our transplant protocols widely within the US, and I would say globally as well.
Thank you. Do you think?
Time
go ahead,
to
Dr.
answer a few questions in the chat, or do you want to take that on?
Sure, go right ahead.
So there was a question, any role for screening APOL1 in Asian patients? And there has been no reason to think that APOL1 variants are present in patients who are not of sub-Saharan ancestry, and therefore there would be no reason to screen at the present time. And there was another question, does the Renocyte panel from Natera adequately capture APOL1 risk variants? I believe it does capture both G1 and G2, but if anybody knows differently, please speak up.
That's what we use in our center, and it does have G1 and G2.
Excellent.
And that's what we're using Natera as well.
Excellent.
And then what do you have set up, and maybe Dr. Nichols is the best to answer this, as far as genetics counseling? Like you said, there's a lack of teaching on our part. I think a hesitancy even in my own practice has been, if I order the test, who will I send the patient to really make sure I am doing the best to give them the health literacy and understanding they need for it. And that's not really there widely. So what do you do, and what can you advise us to do to be sure that we're educating our patients the best we can if we do order these tests?
So currently, what we do is we're sending patients to our genetic testing center where they can counsel patients. But I think that we're sidestepping our responsibility as nephrologists, and what we should do, and I know that you're very much involved in teaching and is a former program director myself, what we really need to do is to begin early, right, within our fellowship programs, to begin training our fellows around a number of different things. One, about disparities that exist in CKD, about genetic testing, but about counseling, and specifically right now there's no standard protocol regarding the elements that are included in counseling. We probably need to put something together regarding that. Not only just the specific test, but when the test comes back, a patient who has 2 high-risk alleles, what is it that we want to tell the patient? Do they have kidney disease that's ongoing, or are they now maybe don't have CKD, but now are at risk? What are the advice items that we want to talk about in terms of lifestyle modification that Dr. Kopp mentioned, blood pressure control, et cetera. But also patients need to be aware that currently there is no FDA-approved therapy, but even so, that they can still benefit from this knowledge, in that they can now become more involved, self-aware, and involved in the care of their kidney disease. So I think that there is utility in putting together a counseling program for nephrologists, and I would add some aspect, maybe minor, for primary care physicians, because they see the majority of our patients before we do. And so if we can start within our fellowship program, then we can expand and continue that throughout, our careers. So that's what I would advocate for.
Thank
Another
you.
question was mechanism of action of the drug. Does anybody have a comment on that?
I can tell you that it is an inhibitor. More detailed information I don't have for you right now.
And are there any questions that we didn't answer in the chat?
So I thank you. Tommy indicated that Medicare, Medicaid covers in full Renasight from Natera, and some commercial insurance partially cover it, which is wonderful to hear. Natera also offers compassionate pricing to those the insurance does not cover, or would partially cover. So this is really good to know. Maybe this is something that we need to include in our educational materials for our fellows, as well as for the rest of us, so that we can better advise our patients.
Very good. And if there are other questions and someone would like to unmute and ask directly, please do as we get towards the end of the hour.
Hello.
Yes,
Hello.
please introduce yourself and where you're from and ask your question.
Right. My name is Dr. Theophilus NIDDK. I practice in Lagos, Nigeria. I've been on GoCom for some months now, and I'm really interested in APOL1 gene study. I asked a question on the chat, but I don't think anybody tried to answer it. We have a couple of a few patients who were biopsied in our center who are FSGS. Some of them are on CNIs, some are on mycophenolates, mofetil. I'm just asking if there's a role that this investigational drug can be used for some of these patients to see how we can retard their progression. That's just my question. Yeah, thank you.
So if I understand you, I can take that one. So at this point, because the drug is still investigation, is under investigation, and we do not have any approval for use, only patients enrolled in the trial have a potential. And remember, it's a double-blinded, placebo-controlled trial. So even if you're enrolled, we won't know what you're getting. So at this point, the short answer is no, but we will know in a few years if there is an effective therapy. But for right now, unfortunately, no.
And
Dr.
Dr.
Narayan
Thank you very much.
Dr. Narayan has asked, what's the mechanism of Vertex drug acts? Do we know?
The exact, so I know that it inhibits the, as I said, it inhibits the APOL1 mediated activity. And let me see, if it does inhibit, I believe it's the cell lysis, but I'll have to don't quote me on that. I actually would need to go back and look at some of the preclinical data.
So I'll answer your question, Dr. Narayan, that you didn't ask, which is there's another drug being developed, which is antisense by Ionis. So there the concept is that the APOL1 RNA is single-stranded like all RNAs. And by introducing an antisense, it binds the sense. And double-stranded RNA is something that our cells do not normally have, but viruses have. So we have a protection mechanism. When double-stranded RNA is present in the cell, it's destroyed as quickly as possible. So again, the antisense by binding to the sense creates double-stranded RNA and it gets destroyed. Now that also is in development. I don't remember exactly where. I don't recall if it started phase 2 or not, but it's encouraging that this has been recognized as a target to develop drugs against. And at least 2 companies are pushing forward with developmental programs and hopefully others as well that I don't know about.
I know as much as you do, Dr. Kopp, and I don't know exactly where they are in the phase of their clinical trials. Some may still be in some preclinical work, but my understanding also it's targeting inflammasomes. So very, very interesting. Dr. Johnson, do you know when the study will begin, or did it already start in recruiting?
We are currently recruiting at sites across the U.S. And we hope to expand to the rest of the world.
Thank you.
With some sites in South America as well as Europe.
And Dr. Lau, I believe, has a question. She had, there was a couple questions, and please unmute and ask.
Thank you so much for your presentation. Dr. Johnson, you mentioned that your interventional study is also recruiting for hypertensive disease in patients with the high-risk alleles. Do these patients with what we think is hypertensive kidney disease, do they need to be biopsied as well? Because you mentioned the FSGS requires biopsy, but do these other hypertensive disease patients need biopsy as well?
So first, let me clarify, to be eligible for the study, you don't have to have a biopsy, but we know that clinically, most patients with a diagnosis of primary FSGS will usually have had a kidney biopsy. So the short answer is no. The patients with hypertensive kidney disease do not need to be biopsied. If they meet the criteria for eligibility, they can go through screening. And if they are selected for the trial, then that's how they can enter. But there is no biopsy requirement.
Can you remind us the criteria needed to enter the study?
So 18 to 60 years old, proteinuria 0.7 grams per gram, as much as up to 10. Non-diabetics with GFR between 75 and 25.
So that set of criteria will allow many patients with so-called hypertensive nephrosclerosis, which is what the biopsy might show if it were done, but we don't usually biopsy them, to be enrolled. So 0.7 grams or greater, that should allow the study to include those kinds of patients.
That's correct.
That's great.
So we're going to capture patients that will have adaptive pathophysiology as well, right? We're not just looking for, nephrotic syndrome, idiopathic or, FSGS primary. So we're going to capture the possibly secondary diseases as well, correct? I just want to be sure I'm understanding.
That is correct.
Okay. Thank you.
And it appears that APOL1 plays a role in both forms, both the primary and the post-adaptive FSGS. So it would make sense that targeting both could be successful. We won't know until the study gets done, but it's a good question.
And one last question and point that had come up in the chat was about preeclampsia is associated with fetal APOL1 high-risk variant and leads to a potential lifetime risk of hypertension. Do you guys have any information or have done any work on any of that? Because there was a question.
I'm not familiar with this. I don't know if Dr. Kopp is about preeclampsia. I mean, I think you can infer that with preeclampsia and high risk of long-standing hypertension, that you may have an increased risk if tested or if you have the genotype, but I don't have any basic knowledge.
Right, the preeclampsia story is important and is modeled in animals as well, that is transgenic mice. But we don't fully understand the mechanism. At least I don't recall that we know where it's expressed in the developing fetus and so forth. But that is an important point.
All right, thank you. Is there any last questions? We will give another.
I think I just want to make one last point that I'm really thrilled to know that we have emerging studies that are, on therapeutics for targeting APOL1. I think this whole topic also raises the question about recruitment and retention of diverse populations in clinical trials in general. I mean, at least in this particular study, we're targeting those individuals who are specifically at risk. So the study is enriched for that, but in clinical trials in general, we have not been doing a very good job about that. And so maybe this also opens another door for us to understand that in diseases that affect underrepresented minorities and other individuals at risk, that the clinical trials themselves should be enriched in terms of recruitment and retention for individuals who are most affected by the disease.
Thank you. Thank you so much. And I think if Dr. Johnson has any comments, we'll end on that.
I have one more comment. I just
go
want
right
to
ahead.
make sure that for the people who asked about the mechanism of the Vertex molecule, it is a small molecule that's an inhibitor for the APOL1, and we think that it stops, as I said, I think it stops the toxic pore formation, the pore formation that leads to cytotoxicity. And this is the mechanism of action. I know that Dr. Kopp talked about another investigational drug that's targeting a different mechanism. And so I think that's in and of itself very exciting that within a few short years, we have now two potential therapeutic targets.
Yes, thank you. I think it's all in all very exciting that we can advance hopefully very soon what we can offer to our patients, individualized medicine, and really tackle a lot of the issues around diversity, equity, and things like that. I think opens a lot of doors for that, and education and health literacy. So I thank you all, Dr. Johnson, Dr. Nichols, Dr. Kopp, for your time today, joining us in this discussion, and all of the participants for their questions and interacting. It's been great, and hope to connect with each of you guys again very soon.
Thank you very much.
Yes.
Thank you.
For putting this together.
Yes,
Thank
thank
you.
you all. Have a great rest of your day or evening.